Yeah, this is the part I don't really like.
A sealed cave, 10,000 bats, no exit, no light, and then silence. What the scientists found inside weeks later would stun even the most seasoned researchers. Why seal bats in a cave? Rwanda, 2024. Deep inside a mining tunnel in the country's southern district, a colony of Egyptian fruit bats became the unlikely stars of a serious health scare, the Marburg virus outbreak. Scientists had been monitoring these bats for years, but things took a dark turn when the virus suddenly made the leap from animals to humans. According to the World Health Organization, one person became infected and died. That marked Rwanda's first confirmed Marburg case ever. So why were researchers so focused on these bats? Well, Egyptian fruit bats are known reservoirs of the Marburg virus, and these tunnels gave scientists a perfect live lab to study how the virus survives and spreads in wild bat populations. See, bats are mysterious animals. They carry viruses like Marburg, Ebola, Nipah, and even variants of the coronavirus. But somehow, they never get sick. That's exactly why researchers are wildly interested in them. And so to understand how these viruses spread, especially before they jump to humans, scientists created what they call a closed system. They found a means to temporarily lock the entire cave so no bats can enter or leave. Just the colony inside, with their viruses. First, they sealed the entrance using mesh or gates. This kept the colony contained and prevented other bats from flying in and altering the results of the data. Then, they used nets to keep them within a space, and attached lightweight GPS or radio transmitters to track movement and social contact. This helped the researchers understand their communication systems and who interacts with whom, because bats essentially see with sound, not light. They emit rapid ultrasonic clicks that bounce off walls, obstacles, and even insects. Those echoes return to their ears, and their brains translate the timing and frequency changes into a mental map of their surroundings. Let's rewind to the first real experiments in the 1940s, when Donald Griffin and Robert Galambos suspended bats in an enclosure with wires. When the bats had their mouths or ears blocked, they crashed, but when unobstructed, they zipped around effortlessly. Griffin even coined the term echolocation in 1944 to describe this phenomenon. In a dark cave chamber loaded with obstacles, bats would switch gears based on how cluttered the environment is. In tight spaces, they emit short broadband calls to get precise echo timing and avoid collisions. In open spaces, they favor longer narrowband calls that cover more distance. But of course, this setting was entirely new, and this seemingly new environment was beginning to get uncomfortable for the animals. The bats began to show signs of stress. Research showed that the bats reacted to stress through something called the HPA axis. This system tells their bodies to release stress hormones like cortisol or corticosterone, depending on the bat species. In another standardized restraint study using big brown bats, researchers found that after just 60 minutes of physical restraint, their stress hormone levels shot up fast. At the same time, their testosterone levels dropped, which means stress can also affect their reproductive system. Now think about what that would mean in a dark, unfamiliar cave. The bats were deeply confused and on edge. Their heart rate spiked, and many of them went into panic mode. This natural reaction caused them to freeze and stay completely still, hoping not to draw attention. Some even began calling out with distress sounds, which most certainly triggered panic in other bats nearby. In fact, another study showed that when bats hear another bat's distress call, they freeze too, with heart rates jumping in response. And just like people, bats can actually remember stressful events. One bad experience in a cave could make them more anxious the next time around. However, there was no escaping this. It was crucial for research as scientists needed to examine how these creatures contributed to disease transmission. They had to collect blood, saliva, urine, feces, and tissue samples over time to check for active viral infection and shedding patterns. In fact, other captive bat infection experiments in lab settings confirmed bats remain healthy, but shed Marburg virus in bodily fluids for days. But this particular experiment revealed something that the scientists weren't ready to find out. Their suspicions were indeed true. The virus could spread bat to bat through grooming, biting, or body fluids. Just as they feared, direct transmission is possible. They also discovered that bats shed infectious virus onto fruit, guano, or cave surfaces. One study found that Marburg virus remains viable on fruit up to 6 hours, meaning humans can get infected by handling contaminated fruit. At some point, they had to begin tracking exact bat movements to allow scientists pinpoint where and when humans might encounter infected bats or fruit and how to avoid them. This was unlike anything they'd seen, unlike what they'd found after the deadly coronavirus outbreak.
They're coming quick. Here they go.
This is not like any cave I've been in. Bats usually try to avoid you. It almost seems like they're coming after us.
So, in 2023, after COVID, scientists began digging deeper into bats to understand the zoonotic risks. That is, the chances that viruses jump from bats to humans. One standout study came out of University College London in mid-2023, where researchers analyzed bat poop samples from UK bats to look for viruses, including coronaviruses. For over 2 years, they safely collected fecal samples from 16 bat species and sequenced them. They found 9 complete coronavirus genomes, including 2 new types and 4 sarbecoviruses, the same group that the SARS coronavirus variant 2 belongs to. The researchers then created safe, lab-made pseudoviruses using the spike proteins to test entry into human cells. One of the sarbecoviruses could bind human ACE2 receptors, but only in cells engineered to overproduce those receptors, which isn't what you'd find in our bodies. This means that none of the viruses found in UK bats could infect humans yet. Now, this new project had shattered everything they thought they knew. The closest they had been to such a discovery was when another important line of research came from the U.S. Geological Survey. They had been doing experimental infection trials with North American bats, asking, can humans pass SARS-CoV-2 to local bats? Initial trials showed that big brown bats resisted infection, but Mexican free-tailed bats were susceptible. However, the studies were pending as of mid-2023, and it was never determined whether they could pass it to humans. But now that they had succeeded in determining the process and mode of disease transmission, they were about to find out what went down in the sealed cave. What happens in total darkness? We know more than anyone that bats aren't loners. They do live in colonies, but only if there's enough room for each creature to socialize. Take the Montfort Bat Cave in the Philippines, for example. That cave holds over 1 million bats, and scientists were shocked to find that the bats weren't exactly getting along. They were flapping and tussling for wing room. Meanwhile, in the less packed zones, some bats were mating and forming social bonds. In this case, overcrowding indeed made them aggressive. Wing jabs, squeaks of protest, and even scuffles became more common. This phenomenon is what researchers call a behavioral sink, where animals break down socially under pressure. Movement was extremely tasking for these creatures, but communication was even harder because bats rely on echolocation to move around. But when they are making random sounds and calls all at once in a sealed cave, their sonar signals overlap, clash, and jam. One study even found that when bats were released into a tunnel to simulate cave exits, their chances of escaping cleanly dropped from nearly 100% when alone to around 60% when packed together. Another study on their group flight behavior confirmed the same thing. Bats are smart enough to avoid crashing most of the time, but only if they can spread out as they fly. Lock them up in a cramped cave with no way to space out, and the confusion just multiplies. But unfortunately, that's exactly what happened here. Now at this point, they began to wonder if they would eventually form a hierarchy or social structure, and they found out that it all depends on species and context. In most cases, bats don't form a rigid hierarchy the way lions or baboons do. The vampire bat, for instance, is one of the best studied species, and they are often used as ground zero to major bat experiments. Now, researchers observed about 1,000 food-sharing encounters among 33 adult female and juvenile vampire bats. They found that female dominance ranks were mainly random. In essence, older or bigger bats didn't consistently win, and higher-ranking bats didn't always win contests over food. Any female could beat another, regardless of status. Male vampire bats did compete with each other over territory, preferred roosting spots, and access to females, but females mostly depended on grooming and food sharing to build social bonds, not for hierarchy.
But plan to get up early. The bats like to be inside by 6:30 a.m., so be there by 5. The Bat Flight Breakfast began in 1957 as a way to encourage visitors to see the bats in action. As you know, they are night lovers.
Some bat species also show spatial structure, where bigger or older individuals occupy better spots near cave ceilings or deeper inside roosts. That may look hierarchical, but it's more tied to resource defense than formal leadership. For example, Jamaican fruit bats exhibit resource defensive polygyny, meaning that dominant males stake out small cave patches while females pick the best ones. Likewise, in leaf-nosed bats and others, we see higher-ranking individuals claiming sweeter spots, but maternal kin and social bonds matter more than absolute dominance. In many bat societies, cooperation trumps dominance. Vampire bats groom and feed each other, care for the pups, babysit, and the females build networks of trust, not dominance chains. Even colonial species like Egyptian fruit bats spend their days sleeping together, vocalizing and squabbling lightly over food or space. Ultimately, their order comes from who they trust, where they fit physically in the group, and how often they cooperate. However, there was one thing that these researchers feared would happen that could completely ruin their experiment or whatever social structure that the bats had going on.
A fungus that could cause a deadly disease among bats was just found in Carlsbad Caverns.
An outbreak of the white nose syndrome. The white nose syndrome is a deadly fungal disease that affects hibernating bats. It is caused by a cold-loving fungus called Pseudogymnoascus destructans, which grows on bats' skin. It's visible especially around the nose, ears, and wings while they're in their hibernation. What happens is when bats hibernate, their body temperature drops and their immune system slows down. The fungus takes advantage of this, growing on the bat's skin, and this results in waking it up too often during hibernation. Well, this doesn't in any way appear fatal, but bats rely on carefully rationed fat reserves to survive the winter. Every time they wake up from hibernation, they burn energy. And if they wake up too many times, they run out of fat, leave the cave too early, and often die of starvation or cold exposure. Since it was first discovered in New York in 2006, the syndrome has wiped out millions of bats across North America. In some caves, it's killed over 90% of the local bat population. Entire species, like the little brown bat, have been pushed toward extinction in some regions. In the case of these locked-in bats, they would rub against each other constantly and wake more often during torpor, which will wipe out fat stores quickly. Arousal frequency skyrockets, dehydration kicks in, blood becomes acidic, and electrolyte imbalances build up. The more they wake, the less energy they have. Some won't even make it out alive.
The fungus that causes it was identified in Texas, and last week, the disease was confirmed in Oklahoma.
Such high density also means more skin-to-skin contact. Since the fungus spreads primarily by direct contact, an overcrowded cave like this one could turn into a transmission hotspot. Even if only one bat starts infected, it can quickly spread to nearly all others. Plus, the cave walls and soil act as a reservoir, harboring spores that persist through seasons. So even bats that don't physically touch infected individuals can still pick up the fungus from the environment. Unfortunately, certain species like little brown bats, northern long-eared bats, and tricolored bats are particularly vulnerable to this subtle but dangerous disease. Soon enough, the researchers knew that the bats would have to make a choice between mayhem and adaptation. Would they spiral into chaos, turning on one another in desperation? Or would they find a way to restructure their colony and carve out new sounds, new systems of survival? Chaos or order? When bats get packed tightly, like in Montfort Bat Cave, their behavior turns somewhat violent. There was more wing flapping, squabbles, and restlessness in the densest zones. Now, this is where food sharing becomes a focal point. Scientists needed to find out what would happen when the bats get hungry. Among most bats, especially insectivorous or fruit-eating types, sharing meals is almost non-existent. They forage in isolation and don't regurgitate food to others. Vampire bats, on the other hand, are a bit more generous. They can't afford to miss meals because they die after about 60 to 72 hours without food.
Based on insects, mainly moths and flying beetles.
So if a bat comes back hungry, its roost mates, especially females, will sometimes regurgitate blood to feed it. Mothers will feed their pups, but adult females also help each other. But in this dark cave, sharing food seemed to happen based on reciprocity. Bats preferentially began to help individuals who have helped them before, or at least who groom or otherwise interact often. In controlled experiments like this one, whether two bats shared food was best predicted by how much food they had already exchanged, not whether they were kin. Donor bats are more likely to initiate sharing than recipients begging, but only with familiar, frequent partners. If a bat never shares, others eventually refuse it when it's in need. But what if that happens, and a bat has to resort to eating another of its kind? These researchers were curious to discover what the desire for survival could make these creatures do. Well, some bat species are indeed known to eat other bats. For instance, greater false vampire bats have been observed feeding on peers in the wild, and in maternity colonies of greater mouse-eared bats, researchers have documented rare cannibalistic events. But these are merely exceptional cases. Cannibalism in bats isn't a typical response to overcrowding. It's a species-specific predation behavior. If food becomes deathly scarce inside the cave, bats will fast. Most species can survive days to weeks without a meal by lowering body temperature and slowing metabolism. They don't automatically gnaw on each other out of hunger. Unless it's a species like the false vampire bat or an extreme survival scenario, cannibalism doesn't become a default. Yet, these researchers feared something even worse, a death spiral.
The volume of 747 jet airliners every night. So think of the ecology and how wonderful.
This is a flight error where bats circle so tightly they fly themselves to exhaustion. Bats can and may do this, but it's not a group catastrophe. It's more about orientation mistakes during swarm emergence, not overcrowding. Scientists eventually came to realize that in a locked cave, bats would likely stop flying non-stop. They'd roost and conserve energy. As a matter of fact, after all the noise and tension, the colony slowly started to adjust because bats are way more adaptable than they look. The bats began to settle into roles and zones. Some move to the edges where it's cooler. Others find tighter crevices to hang quietly. Researchers at Montfort Bat Cave saw this firsthand. Initially, the bats in crowded areas were more aggressive, flapping nonstop. But after a little bit of time, they even began mating. Echolocation also became more structured and exact. The bats stopped shouting over each other with echolocation. They just began to tune it. In the dense caves, they began to tweak the frequency and timing of their calls to avoid colliding into the calls of their mates. Even if the cave was packed with toxic ammonia, they would develop ways to handle it. Their bodies would filter it out better than other animals, and they even create swirls of airflow in flight to cool the cave and reduce gas buildup. Now, the experiment was finally over. They had collected their data and gathered their findings, but would the creatures ever remain the same? Opening the cave. After the bats were confined for 21 days, they were finally released into the wild, and the aftermath wasn't quite what anyone was expecting. After the release, there were a series of noticeable changes in the behavioral patterns of the bats. Captivity, even temporary, introduces acute and chronic stress. Various studies on big brown bats show short-term restraint can elevate stress hormones like corticosterone and cortisol, which can rapidly suppress reproduction and alter behavior. In the 21-day period, the bats went through a range of stress responses, from heightened alertness and pacing to possibly depressed activity levels and erratic social interactions. Some of the researchers thought the bats would go berserk and destroy everything, but that is highly unlikely because bats aren't prone to chaos or destruction. Instead, their reactions were more subtle. They either shifted roosts, changed foraging patterns, or adjusted flight behavior as they recalibrated after confinement. In studies of hand-reared bats released into the wild, researchers tracked normal nighttime foraging and commuting behavior within days, indicating adjustment, not breakdown. One study involving pipistrelle bats, hand-reared in large flight cages for up to 21 days, showed 70 to 90% of them survived at least 4 to 14 days post-release. A similarly designed Italian experiment found 19 out of 21 bats displayed typical roosting and foraging behaviors shortly after release. So they adapted, even after weeks in confined conditions. They also seem to vocalize more, not less, as they navigate a new environment and set up a perfectly coordinated re-emergence of social structure. For the first few days, their flights were altered. They began to observe unusual social clusters or separation, and their foraging attempts increased at odd hours. But once they regained orientation and stamina, they began settling into more predictable routines, normal foraging, consistent roosting sites, and smoother social interactions. What it taught us. Now, scientists had seen firsthand what a bat colony could do in isolation and darkness. They moved fast, never crashed into each other, and somehow could tell what the rest of the group was doing. That is what scientists call swarm behavior. And it's an interesting discovery because it's exactly what people building artificially intelligent technology are trying to copy. But why bats? It's because these creatures are masters of flying in the dark. They don't rely on their eyes. They use echolocation, which means they make clicking sounds and listen to the echoes bouncing off objects. No matter how large the colony may be, they don't bump into each other. They fly in sync, and they make decisions as a whole. If we can understand how they pull this off, we can teach robots to do the same, especially drone swarms that need to fly through tight, tricky spaces. Because if bats can adapt in a cave, robots can be trained to do the same in a collapsed building, a tunnel, or even deep space. Some researchers, like those at the University of Konstanz, are already building robots that mimic bats' echolocation. Others, like Dr. Yuan Lin from Virginia Tech, studied how bats change their sonar when flying close together. That data helps build better swarm robots that know when to speak up or stay quiet. Another group, Suarez and Iglesias, built a robot swarm that explored unknown spaces using what they called a bat algorithm. And it worked. The robots moved through dark, twisty areas by copying bat behavior. Besides the AI swarm logic concept, this controlled research also helped to better understand pandemic threats. Bats are known to host a huge variety of viruses, like coronaviruses, paramyxoviruses, filoviruses, rabies-related lyssaviruses, and more.
These bats are perhaps the one Himalayan animal that deals with extremes by avoiding them.
Their immune systems let them carry infections without getting sick, while they shed viruses into the environment through saliva, urine, and feces. But by confining these bats in a controlled cave setting, researchers could monitor how much virus is being released over time, how long virus particles linger on cave surfaces or in air, and how likelihood of spillover changes with density, stress, and seasonality. That matters because viruses in bat colonies can persist long enough to infect other wildlife, or even people, especially if a cave is visited or disturbed later. These scientists had found exactly what they were looking for. If confinement shows shedding spikes during specific times, health authorities can plan targeted surveillance around those peak windows. Lessons from captive settings such as this one can help build guidance for limiting human-to-bat contact, decontaminating caves, or even restricting access during high-risk periods. In extreme cases, conservationists have created artificial hibernacula to monitor and contain diseases like white nose syndrome in North American bats. Similar designs could be used to study viruses while protecting both humans and bat populations. Now, after scientists proved that somehow bats could help us in AI inventions, they went ahead to uncover something rather unexpected: how these creatures could have the same thinking patterns as humans. Just like the bats were stuck together in one space for weeks, no flying out, no exploring, just each other. We could liken it to locking a group of people in a house together for the same amount of time. No phones, no social media, just them. Over time, they start thinking alike, even if they didn't agree on everything at first. That's where groupthink comes in. Groupthink is when people in a tight group start to avoid disagreements just to keep the peace. They stop challenging ideas even when something doesn't feel right, because they don't want to be the odd one out.
If you want to know what a little angel is, Mexican free-tailed bats, they can see in the dark, they can find their own baby.
Normally, bats in species like Bechstein's bats form flexible groups. They break up and come back together all the time. But when they're confined, researchers noticed something interesting. The group becomes tighter, more predictable. Some bats lead, others follow. And if there's disagreement, like which part of the cave is warmer, they usually just go with what the majority prefers. One study found that when bats had to make a decision, and not everyone had the same information, they still went along with the popular choice. And if the group couldn't agree, they'd split into smaller groups instead. This comes across as all too familiar, because humans also feel pressure to agree, even when they don't really want to. Another study showed that when bats in the same group communicate, their brainwaves actually sync up, so tightly that bats outside the group do not share the same thoughts and patterns, so they are ignored. Humans do this too. We pay more attention to people we like or agree with. People outside our group often get tuned out, even if they have good ideas. That's classic groupthink behavior. Nature adapts, even in silence, in total darkness, when surrounded by chaos. What the bats did in that cave was smarter than anyone expected.